speak. So this captures the basic idea of the question I'm looking at, which is you have these different surfaces in which you can think of as Riemannian surfaces or as surfaces embedded in 3 space if you prefer. And the question is, can you try and somehow measure how close some pair are? So this, I think of this as a problem in applied geometric topology. There have been all these methods, very powerful methods developed in 2- and 3-dimensional geometry and topology, and I think they're going to find lots of real-world applications, and I'll talk about some that we've started to explore. And I think particularly interesting is applications in biology, because I think a lot of biological problems turn out to be geometric in their essence, and I'll talk about some of those. So let me start by looking at an overview of the problem. Here's 9 different shapes, 9 different surfaces in 3 space. And what we'd like to do is try and create some kind of similarity to tell which ones are more similar and which pair are less similar. It turns out the human eye is very good at picking out 2-dimensional shape, but it's not so good at discerning the differences between 3-dimensional shapes. So what are some of these? Everybody recognize these 3 medals? There's the Nobel Prize, the Fields Medal, and the this one's from the Institute. I'm not quite sure what that is. So presumably we'd want something which would notice that these 2 are fairly similar, and not as and similarly with the 2 at the bottom right there. So we'll see how to make sense out of this question. But one interesting thing is that this question really arises almost everywhere. Everything we see is a surface. Our eyes see 2-dimensional surfaces. And so this question arises in many areas and has been looked at by different groups independently. So people studying proteins are interested in the shape of proteins. In medicine, people are interested in the shape and growth of tumors or the shapes of brain cortices. And facial recognition is another problem where you want to try to compare how similar 2 surfaces in 3 space are. And we're generating a tremendous amount of geometric data now on these things, but there aren't we still don't have very effective tools to compare surfaces in most cases. So what our goal will be is to create a metric on the space of surfaces. And I'm going to stick for now to spherical surfaces, surfaces of genus 0. And we'll be looking for some function that takes 2 such surfaces. So we'll look at the set of surfaces up to isometry as the elements of our set, and we'll take 2 such and assign a distance to them, which satisfies the 3 properties of a metric. And it turns out that these 3 properties have very important implications for applied problems. For example, the first property that says that two surfaces have distance 0 if they're isometric, that says that we're looking for an intrinsic measurement of distance. So, for example, if we were looking at faces, we would get distance 0 even if the face is in a different configuration, frowning or smiling or so on. If it's the same surface, intrinsically, you'll get distance 0. So, for example, these 2 spheres might have distance 0. We're not looking at the way the surface sits inside 3 space. Symmetry just means that if a doctor comes in and compares a tumor to a sick tumor to a healthy tumor, or a sick organ to a healthy organ, then the distances won't be dependent on the order of measurement. And the triangle inequality says that the measurements aren't sensitive to noise. So if you have two surfaces that are very close, then the distance from a third surface will also be in close alignment. So these are useful properties to have. And besides a distance, besides a number that tells us how similar two shapes are, the other thing we really want in many applications is an actual correspondence between the two surfaces. So if we have two brain scans, we'd like not just a measure of a number that tells us how similar they are, but actually a diffeomorphism from one to another, an alignment of one to another. So we can, for example, see if one brain is different from another, what part of the brain is different. So as a warm-up to what's been done and what we've done in 3-space, let's go down a dimension and just talk about regions in the plane. So I took some local regions in the plane and some not so local. So the human eye can tell pretty quickly which ones are more or less similar, although sometimes it's not completely clear. It depends on what kind of features you want to measure. So probably the local people understand what these are. If there's any graduate students, this was a singer in the 50s. So I don't know if people still recognize that. So some of the applications of two-dimensional shape comparison are character recognition. You scan a character and try and say which of the 26 alphabetic characters it's closest to. Facial recognition from two-dimensional photographs, fingerprints, so on. These are all these are all essentially problems of saying how close two-dimensional shapes are. This computer vision problem is an interesting one. Just trying to tell whether a photograph shows a cat or a dog was a real challenge in computer vision. As of about 5 years ago, the best algorithms were right under 60% of the time. And guessing would give you 50-50, right? So it's not too much better. And you can see it's quite difficult with some of these, right? But Google and Facebook and so on have put a lot of effort into this problem. And actually, this was a proposed Turing test for a long time. It was something that humans can do very well. But the best algorithms now are over 80% correct. So there's some significant progress being made on this. As far as I know, the real mathematical theory was initiated by Mumford for shapes in the plane, and he wrote a very interesting paper in '91 in which he studied various types of metrics on plane shapes and compared them with human perception, which is also a way of trying to decide. You can do various surveys and try and figure out what looks the same. So I won't go through all of these, but briefly, the Hausdorff metric, you have two shapes, for example, this square and this other square, but this square has a little noise when it was scanned, so there's a little dot to the right there. So the Hausdorff metric says try and align these two shapes as best you can, and then take the distance of one point of a point in one set from the maximal distance of a point in one set from a point in the other set. The Gromov-Hausdorff metric allows re-embedding the shape in higher dimensional Hilbert spaces. But this particular metric has this problem with odd points of noise. One attempt to correct that was the template metric, where you measure the area of non-overlap after optimally aligning the surfaces. So that didn't get affected much by points, but it would, for example, show that this shape, which has a long gash in it of small area, is very similar to a square. That might or might not be something you want. Recently, people have been putting a lot of effort into looking at optimal transport metrics, in which you think of each of these regions as a pile of sand and ask how much work do you need to move one pile of sand to another, and that turns out to give you a distance between the two shapes. That runs into sort of problems when you have when you have a shape like this and you want to move a little bit from one corner to another corner, the distance can be quite large, even though you might want to think of it as small. The optimal diffeomorphism looks at how much energy it needs to stretch one shape onto another. The problem is, again, that you often have artifacts and tears in objects. So Mumford also studied something called maps with tears, which are optimal diffeomorphisms allowing some tearing. So these were all led to a variety of different metrics, each appropriate maybe for some different problems. They all have drawbacks. For example, maps with tears was very hard to compute, it turned out. It was known to Mumford's students as maps with tears. So I want to go off track a little bit just to tell you about some experiments Mumford did, which don't really have that much relevance to the rest of the talks, but they're really fascinating. And they show that mathematicians can really do real experiments, which is sort of unusual, I think. So Mumford worked with some psychologists at Harvard and took two groups of subjects and with two different groups of subjects created a distance matrix, a perception-based distance matrix for these 15 shapes. These are 15 different regions in the plane, and the two subjects that were tested were one a group of pigeons, and the second was a collection of Harvard undergraduates. And the way the tests were run was so a pigeon, one of these shapes was assigned to a pigeon, and a pigeon was trained over the course of some time to peck at a key and get a reward only if that shape came up and not if one of the other shapes came up. And by measuring the number of mistakes the pigeon made, you could get an idea of how similar in the pigeon's brain two shapes were. So from that data, a distance matrix was created for these 15 shapes. For the Harvard undergraduates, slightly different, they measured reaction time. A student was told, only press a button if shape M comes up. And then the reaction time was measured for the different shapes and how long it took them to recognize that it was shape M rather than some other shape. And what was discovered, so the results were consistent across both experiments, and they found that in both cases there's a very large non-symmetry in the metrics. So for example, if you taught a pigeon to recognize this shape, it might make a lot of mistakes when this shape comes up, but if you taught it to recognize this shape, it wouldn't make so many mistakes when that shape came up. So the conclusion from those experiments was that human perception may not reflect an underlying mathematical metric. Okay, so that was some history in two dimensions. Now I want to talk about three-dimensional shape comparison. And so our goal here, we're going to restrict to genus 0 surfaces. So given two genus 0 surfaces, our goal is to find some function that says how close they are, some real number that says what their distance is. And secondly, so it should be 0 if they're isometric and otherwise give a measure of their difference. And then we'll also want to be able to find the diffeomorphism that gives an optimal correspondence. So why is this difficult to do? You might think that you could start with some rough correspondence and then create some measurement of distortion and decrease that through some kind of flow until you achieve an energy minimizer of some sort, and then you compare to the energy of an isometry. This has been done. This kind of thing is fairly standard in applied math. But the reason it's difficult is that the space of diffeomorphisms between two surfaces is a very large space. And when you take any computable flows on that space, you run into lots of local minima. So you get stuck, and there's no reason why you should arrive at a global minimum. So people have tried all kinds of ways to get around this, but it generally leads to arbitrary and inconsistent results. So what we're going to do to get around this problem of the space of diffeomorphisms of a surface to itself being so large is we're going to work in a smaller surface, namely the space of conformal maps. So most of you know what a conformal map is, but let me just give a quick reminder. An isometry is a map that preserves both angles and distances. And conformal maps only preserve angles, but not distances. And a famous conformal map is the Mercator projection from the Earth to the round surface of the Earth to the plane, which is the map we often see when we see maps of the world. Does anybody know why the Mercator projection succeeded all other maps of the world? There's an interesting historical reason why. There's many other possible ways of projecting a round sphere to the plane. But the reason this one became so successful is it's the right one to use for navigation. It correctly gives you bearings when you're navigating. So if you go northeast on this map, then the direction indicating northeast or southwest and so on a conformal map is correct. Angles are preserved, which means that bearings are preserved. And that's one of the reasons this became the dominant map. So we want to use conformal maps in our problem of comparing two genus zero surfaces. And here's 5 examples of genus zero surfaces. And so the question arises, can we even find a conformal map from one genus zero surface to another? And then here's the first deep mathematics that we're going to use. The uniformization theorem of Poincare and Kibis says that given any genus zero surface, you can find the conformal map from it to any other smooth genus zero surface. So if you have a surface like this and a surface like this, there is a conformal map from one to the other. Let me illustrate this with the UC Davis perspective. So UC Davis started out as an agricultural school. And the uniformization theorem tells you that there is a conformal map from this genus zero surface to a round sphere. So there you go. So that's the first mathematical theory that we'll use. Now we're going to actually try and do computations with this theory as we develop it. So it's not enough just to have a theory for smooth surfaces. We want to be able to actually compute. And typically, when you have renditions of real-world surfaces, they're given by triangulations. So when you see pictures of brain surfaces or proteins or something, typically those surfaces have 100,000 triangles representing them. So what we need is some version of this uniformization theorem that works for triangulated surfaces, what's called a discrete conformal map. And the theory of these has been extensively developed over the last 30 years. Probably the first result I know was due to Thurston, who showed that you could use circle packings to approximate smooth conformal mappings. And then there's various other approaches you can take with finite element methods or discrete Dirichlet energy, Ricci flow. So by now there have been many methods developed, and some of them have been implemented in software. We actually implemented this recent method that was proposed by Bobenko, Pinkhall, and Springborn, which is very computationally efficient. So the point is that passing to the smooth, from the smooth to the triangulated setting has been extensively worked out. And now I'm going to illustrate our method for how to align two surfaces, and I'll do it with 2 real examples. I'm going to try and show you how to align these 2 brain surfaces. So these are actually 2 brains that come from a database at UCLA. There's no particular reason why we picked brains as opposed to some other surface, except that the brain community has generated a lot of data which they've made available. So it's more the availability of these brains rather than their particular suitability for this method that makes them the first thing we looked at. So these branes are these fairly complicated, convoluted Riemannian two-spheres. They've sort of been arbitrarily sealed off at the bottom, the side you can't see where the spinal cord comes out. And these surfaces are available. And then what you'd like to do is somehow measure how similar two of these branes are. And one way you can do that is you can study these valleys in the brain. These are called sulci. They have importance to neuroscientists. Each one has a name and separates these regions where things like the visual functions or the motor functions will take place. And they're not exactly the same between different people, but there's a high degree of alignment. And so by measuring how well you can align a sulcus in one brain with a sulcus in another, you can measure how good an alignment scheme is. All right, so what we're going to do is start with these surfaces were generated by MRI data, which are shown at the top here. And there's some software that the brain community has to take these various slices and figure out what the edges are and to reconstruct this, a triangulation of this brain from the MRI data. So how are we going to map one brain to another brain? Well, the first thing we'll do is find the conformal map to the round sphere, an angle-preserving map to the round sphere. And we do this using these discrete conformal methods. So we did this. This shows the process for brain one. There's about 100,000 triangles here, and you see them laid out in the plane here. And this map here is stereographic projection. So there's about 100,000 triangles in that black center disk there. And this composite map is a good approximation of a conformal map from the brain surface to a round two-sphere. So we do that for each of the two brains. And then we want to map this brain to this brain conformally. So all we need to do is pick a map from one sphere to the other sphere, a conformal map from one sphere to the other sphere. And the composite will be a conformal map from the first brain surface to the second one. And so we have a choice here. Well, here we had a choice as well, but it doesn't matter what we chose. But here we can reduce the choice of maps from surface 1 to surface 2 to how we choose this Mobius transformation. There's this 6-dimensional family of Mobius transformations. And together they give all the conformal maps from this surface to that surface. So what our problem is which Mobius transformation gives the best map from surface C1 to surface C2. Well, C1 is the surface. From brain 1 to brain 2, C1 is the map. And the way we choose the best Mobius transformation is we'll define what we call the symmetric distortion energy, and the formula is here. So this is an energy which measures the deviance from isometry of this map. And in a way, this is the most natural thing you can do. So we've restricted, we're trying to find the best angle-preserving map, the one that's closest to an isometry. And the idea is, if you preserve angles, how can you measure how far you are from an isometry? Well, at each point, what you're doing is uniformly stretching by some factor, which is what, going this way, you would be stretching by factor lambda f, the conformal factor. So if that was an isometry, then lambda f would be 1. You'd be stretching by a factor of 1. So we just take 1 minus lambda f squared and integrate that and do the same for the inverse map. And if you add them together, you'll get some measurement of how far your conformal map is from an isometry. This is, I would claim, probably the simplest way to measure that. And this energy, the symmetric distortion energy, is what we'll use to give the distance between two surfaces. And so the theorem that comes out of this idea is that if you do this process, then you can prove that there does exist a map which minimizes this symmetric distortion energy. And this isn't completely obvious because you're minimizing over PSL2C, which is a non-compact space. But it turns out that the energy goes to infinity as you leave compact spaces of PSL2C. And the second thing that you can check for this formula is that you get the three properties of a metric. And so the first two are actually built into the definition, but you have to check the triangle inequality holds, which is not obvious at all. So I won't tell you about the proof. It's on the archives. But what I want to tell you is how well this does in actual applications. How it does in distinguishing biological data sets and getting information that's interesting to biologists. So what might we hope if we put a metric on the space of shapes? The hope is that we get something that's sensitive to changes in shape, not sensitive to noise, to small fluctuations of random type, and that doesn't depend at all on parameterization. It should be independent of how we represent the surface, as long as it respects the geometry of the surface. So I'll start by three mathematical experiments, and then I'll tell you about some biological experiments. So the three mathematical experiments. So this experiment says, what is the distance in this symmetric distortion metric? What is the distance to a round two-sphere? So we've taken a family of ellipses which are squeezed along one axis. So the axis the x-axis here measures the amount of squeezing along the x-axis of an ellipse. So 10 to the 0 would represent a round sphere. Each of the principal axes would have the same size then. And then as you go to the right, you're pulling a sphere out to be cigar-shaped. And as you go to the left, you're pushing it down to be disc-shaped until eventually it squeezes down into a two-dimensional plane. So it's sort of shown above. And you can see what's happening to the distance. So it's quite sharp initially. So you see the ellipses that are very near to a sphere are actually distinguished quite well from the round sphere. There's a breakdown in the numerics for very high stretching, and that's because we are using a mesh with about I think 1,000 points to represent the sphere. And once the sphere gets too needle-like, it starts poking, its sharp point just starts poking through the mesh. So here's a second experiment. So that was, in case you didn't get the point, that was a success in the first experiment. The second experiment measures noise. So here we've taken a sphere and we've radially perturbed the vertices of a triangulation of the sphere by some multiple of the edge length. So on the x-axis of this graph, you see the multiple of the edge length. And on the vertical axis, you see the distance. So, for example, in the middle here, 10 to the 0, the radial direction of the vertices has been perturbed approximately the same to be well, it's a Gaussian perturbation which has standard deviation equal to the edge length. And you can see that, so that's a fairly sizable perturbation. And you can see that for perturbations that are, for example, half the edge length, well, half the edge length would be up here somewhere, the distance is still very close. So what this is indicating is that this energy function that we're computing cancels out with small random perturbations. And the third experiment involves subdivision. How does this change as you change a mesh? So we're comparing it to a round sphere again, and this says the number of vertices in our comparison. So, for example, if we triangulated a round sphere with only 10 vertices, then we get, well, 12 would be the icosahedron, or 20 maybe, 12. So down here you'd have something like the icosahedron, which is roughly round, but not exactly. And as you take more and more points, in the red graph they're randomly distributed, and in the blue graph they're they're essentially uniformly distributed, you see that the distance goes to 0 and then it stays close as you subdivide. So this is independent of mesh once you get enough points to nicely approximate a surface. This was an experiment that was done on the UCLA brain database. So what we did here was compare our method to two standard methods that are used in the brain imaging community, FreeSurfer and Landmark. And this is actually represents 18 different experiments, these 18 histograms. Our results are shown in the white histogram, and the black and the red are the two other methods, FreeSurfer and Landmark. So in this experiment, lower is better. What this is doing is aligning two brains and then measuring the Hausdorff distance between these sulci in one brain and the image of the sulci of the other brain. So this is a fairly standard way that that community measures the effectiveness of brain alignments. So the moral here is our method did about the same as the two other methods, but the other methods involve human expertise. They have human experts who go in and mark some of these points and tell you where to map this point and where to map that point. Our methods are fully automated. There's no human input that went into making this alignment. It just took the triangulated surface and automatically made it. It's very computationally fast. And also our method is angle preserving, whereas their methods generally aren't. So you might think that we pay a penalty in making an alignment by requiring that angles be preserved. But this shows that there's no significant penalty in requiring the alignment not to be an arbitrary diffeomorphism, but a conformal diffeomorphism. This is another set of experiments. This came out of a study by Boyer, Lippmann, St. Clair, Puente, Patel, Funkhauser, General, and Dobices. This appeared recently, PNAS, 3-4 years ago. And the nice thing was that they made their data set available. So we were able to get the same data that they used and run our method on it. They actually did something similar in spirit, except instead of this symmetric distortion distance that we use, they used what they call the continuous Procrustes distance, which is a metric on shapes based on the Earth movers metric, the Wasserstein metric, optimal transport metric. And I'll show you how the two methods compare. In this study, they had, I think they had something like 110 bones. This is the bone that's right behind your big toe. It's called the metatarsal bone. And it's a good bone to use because it's fairly small. It usually comes complete instead of shattered in fragments. And they had it from 38 prosimians. So prosimians are things like lemurs and associated primates, and also simians, which were 23 New and Old World monkeys, like baboons. So the idea is you take these topological spheres and we'll assign a distance to any pair, and we'll see how well that distance reflects the fact that they're from the same genus or the same family. We'll construct a distance metric. Here's an example of the measured distance from 4 bones. So we have a baboon bone and a lemur bone from so this baboon B and baboon A. So there's 4 bones, 2 along the top and 2 along the bottom. And for each pair, we see the distance of one to the other. And we've also indicated on this figure, if you map lemur B, the bone from lemur B to the bone from lemur A, just the surface actually, the red here indicates the areas where the conformal factor is high. So that's where there's a relatively large amount of stretching. And you can see here, for example, that the lemur bone has distance 0.36 from this baboon bone, but only 0.11 from the other lemur bones. So it is, for these 4 bones at least, it's correctly picking out bones from the same genus. Here's how human observers do the same thing. So there's a field of anthropology called geometric morphometrics, which does this kind of study, in particular for fossils. When you dig up a bone from a fossil and you want to know what species it belonged to, you can't use genetics. There's no DNA on fossils that are 3 million years old. So you have to use geometry. You have to use the shape of the bone to try and see where it fits in the world of various species. So what they'll do typically is have some human expert mark key points. These are called landmarks. So you'll see these landmarks from 2 different papers I got by Googling geometric morphometrics. And what they'll do is they'll measure the distance between these points and they'll create a vector of distances. And then the distance between 2 individual specimens will be the Euclidean distance of those 2 vectors. That's typically how they'll assign a distance to 2 species. And that's considered state of the art. So the difficulty of such a thing is, first of all, it's very slow and expensive. You can think about, even if you just took 6 points, how long would it take to create a matrix of distances? You have to mark the points. There's a lot of human error involved in an inconsistency from one. If a different person does it, or the same person on 2 different days, they may pick a different point for force and get a slightly different answer. So it's error prone and expensive. How does it do compared to the 2 automated systems, the symmetric distortion distance and continuous Procrusty distance? So that the test that's used for this kind of study, the statistical test that's used to see how well these distance matrices work, is called a ROC test, a receiver operating characteristic test. It came originally out of signal processing. And the way it works is, let's say we have, for example, 2 species, a lemur and a monkey, and we have 100 of each. Then we'll have a matrix, 100 by 100 symmetric matrix of distances. And what we can do is line those distances up from smallest to largest, and then we'll take the smallest one and say, is it the same species or is it a different species? Because we know the answer. So this is a way of testing a distance when we know the answer for which species the bone really came from. So if it's the same species, then we'll take a step vertically because we were correct. If it's the smallest and it was the wrong species, we'll take a step to the right. So if our method works perfectly, we'll go all the way up to the top first. All the smallest distances will be correct, and then we'll go all the way to the right. So perfect measure is up to the top and bottom. If we flip a coin, we'll just go on the diagonal. We'll randomly go left or right. If we get everything wrong, we'll go along the bottom and then up. So that's the test that's used. So the number that you get is the area. The more area that you have under your curve, the better your method is for predicting this clustering based on distance. So you see here 4 different methods that are being compared on 3 data sets. The toe bone is the metatarsal up here. The radius is the arm bone at the end of the outside of your arm. And this is some teeth data set. I'll show you some pictures of those in a minute. Our method is shown in red here. For this method, there were 2 different observers, one in black and one in purple there at the top. So you can see that we came in between the 2 observers, and this was the continuous Procrustes method from the Duke test. And so you can see that here the methods do about the same. Here the observer and our method do around the same. The other method does a little worse. So this was quite successful in predicting in grouping these bones. One thing you can do out of this is try to see where a fossil fits in an evolutionary tree. This is an old world monkey family tree, and on the right there is a fossil. It's called a Victoriapithecus. It's about 3 million years old. And it's thought that it lies somewhere up there in the family tree of current Old World monkeys, and that's purely based on its geometry. So we haven't done this particular model yet, but this is the kind of thing that one wants to do. So we were able to take these data sets for the teeth and so on of these primates, and this is reconstruction. This is reconstructions of the evolutionary tree of these species using these 4 different methods. The 2 left ones are done by human observers and marking landmark points. This method here was the continuous Procrustes distance, and this method down here is our method, the symmetric distortion distance. And we essentially had no mistakes for this particular study. The others had a few mistakes, but not too many. So for example, this observer messed up this one family that's shown in green here. It wasn't able to group there were 4, these were the metatarsal bones. There were 4 toe bones from one species, and it broke them up into 2 groups of 2. It also made a mistake up here. One of these species, this orange species here, was stuck in with the colabar monkeys up here. Anyway, the method that was used to recreate this family tree from these toe bones was completely automated. So there was no knowledge at all used. It was just a scan of the toe bones and then a distance matrix based on the symmetric distortion distance. And then it was fed into a program called Philip, which automatically generates trees from different distance matrices. So no expertise, I can assure you of that. This is another biological experiment. This time, this is fragments of bones from the end of the radial. It's the distal radial, which is the outside radial bone of your arm. This time, it typically gets broken off. So these broken off pieces are used. Here there's one from a human, gorilla, chimpanzee, and orangutan. And so this time, the scans wind up being disks instead of 2 spheres. So the first thing we do to make our method work is to cone off to the center of mass of this boundary curve to produce a sphere. The other methods actually used methods that were tailored to a disk. So we were in a handicap for this method because we didn't use the fact that it was a disk at all, and they did. But nonetheless, we were able to do better than the continuous Procrustes distance on this rock test. And compared to the observer, we do better for small distances, not as well for large distances. Then another set of data involved teeth. So same methodology. You take 2 teeth. In this case, one is from a lemur and one is from a tree shrew. And you map both of them to a round sphere and then look for the best conformal map, the smallest distortion among all conformal maps from mapping one of these teeth to the other. And you can see here an example of the kind of distances you get for these 4 teeth. This tree shrew B is distance 0.55 from flying lemur A and 0.25 from tree shrew A. So the interspecies distances are smaller. So that worked out quite well. But initially, we had some problems with this, with some of these. For example, in this example, A10 and A3 come from the same family, and this tooth, these are molar teeth. This molar comes from a different family, Q06. So the A's indicate one family, and the Q's indicate a different family in this database. And we got that the distance from A10 to A13 was 0.38, which was bigger than the distance from A10 to Q06. So that was not what we expected. But it turned out that in this study, they were using both right and left molars. So and a conformal map preserves orientation. So what we did was we threw in the reflection of A10. So the reflection of A10 is a different surface. Its distance from A10 is actually 0.36. But when you threw in the reflection, then suddenly the distances got the right order. And this is an indication of that. You see, this dashed line is the Rock test without allowing reflections, and the upper red line is what you get when you do allow reflections. So this indicates that this has the ability to pick up orientations as well. So I have a few more minutes, so let me talk a little bit about some other types of data. Proteins are a particularly interesting biological data set. And before I talk about them, let me just say a little bit of philosophy. It turns out that lots and lots of what biologists do is really geometry. Whole fields of biology really are just some very specialized geometry. For example, think what a radiologist does. A radiologist tries to understand images, which are really three-dimensional geometric bodies. That's what they spend all their time doing. So really, they're just trying to interpret two-dimensional surfaces or three-dimensional solids. And many, many other geometric problems arise in biology. So I didn't realize this when I started on this project, but it turns out that these proteins, you often see them drawn like this by biologists. This helps them understand the genetic structure and functions and so on. But their geometry is more like this. They're really sort of blob of atoms. And these are obtained by X-ray crystallography. They really can more or less tell the XYZ coordinate of each one of 20 or 30,000 atoms that form this blob. And we can generate a surface that represents the outside. And the function of a protein, a biologist will tell you, is about, so there's not complete agreement on this, but the computational genomics people think about 95 to 98% of a function of a protein is just determined by its geometry. All the rest of it, the electric potentials and which particular amino acids are near the surface and so on, have a relatively minor role. And the way proteins work is two of them come together, like this little red one and this blue one, and they bind to each other, and that either stops something from happening or causes something to happen in biology. And so a common surface area is very important for biologists to understand, which is really a very geometric problem, this protein docking problem. At this point, over 100,000 proteins have had their shape determined in this way, in something that's called the protein database. So there's really a lot of basic geometric problems involved here that need studying. So we studied perhaps the simplest possible problem you could ask about the shape of a protein, which is how round is a protein. So we want to compare the shape of a protein to a round sphere. We were able to get triangulations of 533 proteins from another study that was done by my co-author on this, Patrice Koehl. And so we ran them through this method, and this is a histogram of the distance from a round sphere. This is a different normalization than before. I've left out a lot of details, but one thing we've done is we've always normalized these surfaces so their area is 1. So scaling is taken away from the problem. You can retain scaling, but we just want to look at shape. And often when you get data, you don't have good scaling information. When you get data out of X-rays, you don't often know. You get coordinates sometimes without knowing what units are used. So we're just using shape and not we're projectivizing the scaling, if you like. So what you'll see here is most proteins, this was a group of 533, most of them cluster in a fairly globular shape. This was the most round, the one that's shown here. And these are the two least round. So you can see this is picking up something of this idea is picking up something of the shape of a protein. Let me in the last I don't know. Should I stop early for questions, or and go till the hour? Okay. So let me talk a little about a different approach. So I talked about this method with conformal mappings of two spheres. But there's a different approach you can use, which is based on hyperbolic geometry. And I want to talk a little about this. So how do you compare two shapes using hyperbolic geometry? And why might that be a good idea? So here's an example. When we have a graduate student who wrote a program to compute hyperbolic structures. So he can take a triangulated surface in R3 and actually compute there's a unique hyperbolic structure on such a surface which is conformal to the Euclidean metric. You have to be a little careful because it's not a smooth metric, but in the smooth case there would be a unique hyperbolic metric that's conformally equivalent, and you can extend that idea to piecewise smooth metrics as well, combinatorial metrics, and that's what was done in this example. So we have a way of computing these, and sometimes in biological data you have higher genus. So for example, when you compare monkey skulls, what the anthropologists typically do is fill in the eyes and the nose, but they leave these holes at the ears, so that this is actually a genus 2 surface. And you can try to compare genus 2 surfaces to understand the geometry of these monkey skulls. So my colleague at Davis, Nina Amenta, has been working with some people at the Museum of Natural History here in New York to do things of this type. This is an example of a hyperbolic structure that was computed on this Macaca leonina monkey skull. So this was triangulated with 30 or 40,000 triangles, and then hyperbolic structure was computed, which was conformally equivalent, and then it was laid down in the hyperbolic plane here. The two dark spots here in the middle would correspond to the two teeth, actually. So we did some work on applying this to brain surfaces. Now, brain surface is a topological sphere, so you might say, how can you compute hyperbolic structures on topological spheres? And the idea is to represent the brain as an orbifold rather than as an ordinary surface. So an orbifold is a smooth surface with some singular points where the total angle is less than 2 pi. These are fairly accurate pictures of what a hyperbolic orbifold would look like. This is an octahedron, I think, and this is an icosahedron, and they have nice, smooth hyperbolic metrics, except that there's certain singularities where the angle adds up to less than 2 pi. So what we did was we took brain surfaces, and we made them into orbifolds. We picked out, I think we picked out 28 points corresponding to the endpoints of these sulci. In this picture, the brain has been inflated. So again, this is software that the neuroscience community has. They have software that will inflate a brain so you can see the surface. You can visualize it more easily. So here you can see the white points sort of represent the endpoints of these sulci. And they were marked by graduate students, these white points, and then undergraduate, actually. And then the sulci wind up being these different colors. And so here you have this gives, really for the orbifold structure, all we need is the white points. We assign an angle of pi to each of these white points. You have to actually, when you align two orbifolds, you have to determine a homotopy class, which is given by marking these, the markings are essentially given by these sulci lines. So that gives you a way to align two brains, and we did this for a collection of brains. This is an example. These were two initial brains. These are hyperbolic. They've been cut open along these sulci, and the hyperbolic structure was computed, and here they are in the hyperbolic plane in each case. And then what we did was, to align the two brain, we minimized the Dirichlet energy. So we just took the the harmonic map from one hyperbolic structure to another. Now the nice thing about this process, it may or may not be useful for the biologist. We can't tell until we get it into some sort of form where they can do biological experiments. But what is nice about this method is that it gives a unique correspondence. Because there's a unique harmonic map from one hyperbolic structure to another, you'll get completely consistent results if you do this multiple times. There's no, I talked at the beginning about the problem being that you run into these local minima when you minimize an energy. But when you take this point of view, these hyperbolic energy minimization problems have a unique minimum, the convex problems. So you never get stuck at local minima. And that means that if you based some alignment method on this, you'll always get consistent results. If you go in 1 week and go in another week for a second scan, the results will be consistent. So this was an example of an experiment we did. We mapped 12 different branes onto one brain, and we aligned the 12 by this Dirichlet energy minimizing map, and we saw how the different sulci align. So these 12 different blue curves represent the sulcus corresponding to this black curve here on 12 different branes. Now, I can't tell you whether there's any geometric significance, any biological significance to that, but there could be. And so one of our goals is to try and get a package together that would allow biologists to experiment with such things to see if there's any interesting biological information on these correspondences. So in conclusion, this same basic problem of how to compare two surfaces shows up in lots of areas. And there's powerful techniques of low-dimensional geometry and topology that have been developed that can be applied to this problem. And one of the roles I think that mathematicians can do is by providing this common framework for this sort of thing, they can prevent people from recreating the same theory over and over again. And that's what typically happens. People in one field will try to develop surface alignment methods from nothing and completely reproduce work that's been done already. And finally, there's lots and lots of other geometric problems that are fundamentally geometric, biological problems that are fundamentally geometric that I encourage you to look at. So thank you for listening.
Thank you very much.